So welcome back to another histology video. Today we'll be talking about question 43, which is the peripheral and the optic nerve. So the peripheral nerve are both spinal and cranial, and they are covered by different layers of sheath by the myelin sheath that separate the nerve into different layers or different concentric layers, like the epineurium, the perineurium, and the endomerium, and they contain both axons and dendrites, and each nerve are covered by the Schwann cells, very important for peripheral nerve. And their functions are that they are mixed, so they are motor, sensory, and autonomic in function. Motor meaning that they go from the CNS towards our muscles, so we can voluntarily move. Sensory from our body towards our CNS, and autonomic, like having both parasympathetic and sympathetic functions. So our body's response towards stress or rest situations. And the myelin sheath in PNS is very important because it keeps our axons insulated. So this means that our ions are not able to travel out of our axons, which means that this creates a more efficient impulse. Um, and in PNS, we also have nodes of Ranvier, which are the unmyelinated parts of the axon, and they are rich in voltage-gated sodium channels. Internodes are just another way of saying the myelinated part of the axons, and the Schmidt-Lantermann clefts are basically spaces left behind by the Schwann cells becoming into myelin sheath. And here are just two schemas that you can reference. So going from inwards toward outwards, we start with the nerve fibers and then nerve fascicles, which are basically just bundles of nerve fibers. And then going more outer, we end up with the nerves. But it's important to know the different layers. So starting from the innermost layer, we have the endomerium, which is the loose connective tissue. Over here, it's where the axons are covered by the myelin sheath. And then in the second layer, we have the perineurium, which contains flattened cells or flattened fibrocytes. And as to recall from the video I made previously, flattened fibrocytes are non-functional fibroblasts, and there also contains tight junctions. And in the perineurium, there contains a specialized connective tissue, which is used for the blood-nerve barrier, for nutrition, or removing waste. And in the end, the nerve is covered by the epineurium, which is dense, irregular connective tissue. And I highlighted the vasa nervorum over here because some professors might say it as this, while others might say that they're just blood vessels, but in the end, it's just to exchange any materials with the blood vessels and the nerves. Here is just another schema to put things into perspective so you can see the cytoplasm and the nucleus of the Schwann cell. Here we have a cross-section of the peripheral nerve in Luxel blue. And to recall, Luxel blue stains the highly lipid content of myelin. So that's why it's very blue and why we can see the myelin sheath here clearly. And it also kind of highlights the Nissl substance. And to recall, the Nissl substance are basically just areas of the neuron that highlights only the rough endoplasmic reticulum along with the ribosomes on it. Here is the peripheral nerve and the longitudinal section. Over here you can see the fibrocyte nuclei, also known as resident cells. These two are interconnected or interchangeable terms. And then you can also see the Schwann cell nuclei and the myelin sheath and also the axons. Here is just another slide from the previous video, just to recall. You can see the nodes of Ranvier here. And here is another slide staining in green trichrome. So now nerve ganglia. So nerve ganglia are basically compacted bodies of neurons or cell bodies of neurons surrounded by glial cells of the PNS. So these are Schwann cells and also satellite cells because they act to nourish the cells. Again, they're bundles of nerve fibers too, and they're enveloped by dense connective tissue capsule, very important. And they can be categorized as sensory and also autonomic ganglia. and yeah, so overall ganglias are like circular structure shaped, filled with neuronal bodies and glial satellite cells or glial cells. They act as a relay station basically for nerve impulses. And so with sensory ganglia, they transmit incoming signals, whereas autonomic ganglia, they control outgoing signals. And yeah, so here's just some more information about the nerve ganglia for sensory. Here you can see the staining or a slide for the sensory ganglia, which are in pseudounipolar neurons. And also you can see the satellite glial cells. And the spinal ganglion over here, stained in HE, you can clearly see the different parts of it. So you can also see the pseudounipolar neurons of the dorsal root ganglia, and you can also see the epineurium of the spinal nerve, and also the connective tissue layer with the blood vessels. And then in this slide, we can also see clearly the cell bodies. Again, very important that they are unipolar neurons in spinal ganglion. And in the autonomic, we basically explained the function already, so they can consist of the sympathetic and parasympathetic part, and they are also very multipolar neurons rather than solitary. So this means that they are interconnected in their function rather than working individually as separate neurons. So this basically means that they all work together. That's it. Um, and they are smaller, more uniform than pseudo-unipolar in spinal ganglia, and they also contain a discontinuous layer of satellite cells around the neurons. Um, Here is just another slide of the autonomic ganglion stained in HE. Another thing that's eye-catching you could see is that there is a lipofuscin, and it's not uncommon to see this in autonomic ganglion. And just to explain, lipofuscin are basically the byproduct of lysosomal digestion in cells that lived for a long time. So basically indicating that, you know, they are going through cellular aging, you could say. And then over here, it's talking about the optic nerve. Um, and on the right, you can see that the slide is stained in luxal blue. Again, luxal blue highlights the myelin present in the nerve. And for histological purposes, you need to know that it's covered by three layers of meninges, which are the pia, arachnoid, and the dura mater. Here is just another view of the optic nerve that I took. So you can see the vessels over here, known as the vasa centralis retinae, and you can also see the layers of the meninges, so the dura, the pia, and the arachnoid. Sorry, the arachnoid. And over here, you can see the layers of the retina. So the way that the optic nerve first comes about is through the ganglionic layer, where these axons would then combine to form the fully fledged optic nerve, which then leaves the eye and enters the brain, as you all know. And here are just more slides of the eye. So we have the posterior view of the eye, um, and we have the longitudinal on the left. And we can also see these central retinal vessels that I also showed you in the previous slide over here, which basically supplies the eye and the optic nerve. And also something very important is the lamina cribrosa that I circled in red. And it's the part of the eye that is located at the back where the optic nerve leaves the eye. And it's it's clinically significant because in diseases like glaucoma, this area could be damaged. And on the right, it's basically just a cross-sectional view. So we have the pia, the arachnoid mater as well seen, and the outermost is the dura mater containing the vessels, and clearly in the inside is the optic nerve. And here's just a longitudinal view of the optic nerve where you can see the glial cells and also some septa present, but the professors won't ask you that. And yeah, that's basically it.